Chemical sedimentation is a separation technique that involves allowing suspended particles to settle out of a solution under the influence of gravity or centrifugal force. This technique is widely used in industries such as water treatment, pharmaceuticals, and food processing. In this article, we will explore the different methods of chemical sedimentation and their applications. Gravity sedimentation. Gravity sedimentation is the simplest and most common form of sedimentation. It is a process that relies on the force of gravity to allow suspended particles to settle out of a liquid. The liquid is placed in a container and left undisturbed, allowing the suspended particles to settle to the bottom. The clear liquid is then carefully removed from the top of the container. This method is often used in water treatment plants to remove suspended solids from water. It is also used in the production of wine and beer to remove suspended solids and yeast cells. Centrifugation. Centrifugation is a process that uses centrifugal force to separate particles of different densities. The sample is placed in a rotor and spun at high speeds, causing the denser particles to move towards the bottom of the tube or container. The lighter particles remain at the top of the container or float on the surface of the liquid. Centrifugation can be performed in a variety of centrifuges, including benchtop centrifuges, floor-standing centrifuges, and ultracentrifuges. It is commonly used in the separation of biological molecules such as proteins and nucleic acids. Flocculation. Flocculation is a process that involves adding a flocculant to a suspension to cause suspended particles to come together and form larger particles called flocs. The flocs then settle out of the liquid more quickly than individual particles. Flocculation is often used in water treatment to remove suspended particles from the water. It is also used in the production of ceramics and paper. Precipitation. Precipitation is a process that involves adding a reagent to a solution to cause the formation of a solid precipitate. The precipitate can then be separated from the liquid by filtration or centrifugation. Precipitation is commonly used in analytical chemistry to isolate and identify certain chemical species. It is also used in the production of pharmaceuticals and inorganic chemicals. Electrophoresis. Electrophoresis is a technique that separates charged particles based on their charge and size. The sample is placed in a gel matrix and an electric field is applied. The charged particles move towards the opposite electrode based on their charge and size, resulting in separation. Electrophoresis is commonly used in the separation of proteins and nucleic acids in biological research and diagnostics.